Assalamu alaikum. This lecture is the continuation of strain improvement. The technique which we study today in manipulation involving foreign DNA is protoplast fusion. First you should know the meaning of protoplast fusion. Protoplast fusion is a physical phenomena. During this type of fusion, two or more protoplasts come in contact and adhere with one another, either spontaneously or in the presence of fusion-inducing chemical. So you can say that protoplast fusion can take place by two methods: spontaneous fusion and by inducing fusion, that is in the presence of fusion of inducing chemical. After adhesion, membrane of protoplast fuse in some localized area, and eventually the cytoplasm of two protoplasts intermingle. Here you can see this phenomena. First, I will tell you about some method of protoplast fusion, and then I will elaborate the phenomena describing here in the picture. Uh, uh, Method of protoplast fusion. Broadly speaking, protoplast fusion can be classified into two categories: spontaneous fusion and induced fusion. In somatic hybridization, spontaneous fusion is of little significance. The method used for induced fusion can be again subcategorized, which I will tell you later on. First, I will discuss spontaneous fusion, which is the first type of protoplast fusion. In this type of fusion, spontaneous fusion, protoplasts during isolation often fuse spontaneously, and this phenomena is called spontaneous fusion. Some physical contact is sufficient to bring about the spontaneous fusion among the similar parental protoplasts. During the enzyme treatment for the isolation of protoplasts, it is found that protoplasts from adjoining cell fuse through their plasmodesmata to form a multinucleate protoplast. Electron microscope studies have shown that as the cell wall are enzymatically degraded, the plasmodesmatal connection between the adjacent cell are enlarged due to removal of its constriction and the enlargement of pit field. Eventually, the greater enlargement of plasmodesmata allow the entry of organelles into the neighboring cell. Finally, a complete coalescence. Coalescence means joining of small bubble to form large bubble of adjacent cell take place. Spontaneous fusion is strictly intraspecific and give rise to homokaryon. The protoplasts, once they are freely isolated, do not fuse spontaneously with each other. An exception is the protoplast from the microsporocytes of some plant of lily family, where the freely isolated protoplast fuse spontaneously. This type of spontaneous fusion has been used to produce intergeneric fusion. For example, the spontaneous fusion of microsporocyte protoplast of Lolium longiflorum and Trillium Trillium species, etc. This is all about spontaneous fusion. In this type of fusion, fusion is taking place spontaneously by involving some physical phenomena taking place inside the cell. But if we talk about induced fusion, what we see in induced fusion, fusion of freely isolated protoplasts from different sources with the help of fusion inducing chemical agent is known as induced fusion. Normally, isolated protoplasts do not fuse with each other because the surface of isolated protoplast carries negative charge around the outside of the plasma membrane, and thus there is a strong tendency for protoplasts to repel one another due to their small charges. So, this type of fusion need a fusion-inducing chemical agent or a system which actually reduce the electronegativity of the isolated protoplast and allow them to fuse with each other. Actually, in induced fusion, it is a highly important and a variable chemical technique because the protoplasts from a widely different and sexually incompatible plants can be fused by this procedure. This is the importance of this technique. This technique has the possibility and ability to combine different genotypes beyond the limit imposed by sexual process. The fundamental objectives of somatic hybridization are mainly based on induced protoplast fusion. The isolated plant protoplast can be induced to fuse by three ways. What are three ways in induced fusion are? First one is mechanical fusion. In this process, the isolated protoplasts are brought into intimate physical contact mechanically and under the microscope using micromanipulator and perfusion micropipette. These are the instruments which are basically used in mechanical fusion. The micropipette is partially blocked within 1 millimeter of the tip by a sealed glass roll. In this way, the protoplasts are retained and compressed by the flow of liquid. By this technique, occasional fusion of protoplasts has been observed. It means this method is not so reliable for the protoplast to fuse. The other method is chemofusion. Spontaneous fusion of two or more adjoint several chemicals have been used to induce somatic protoplasm is of a number of practical use, but protoplast fusion, in what we see in the chemofusion, sodium nitrate, this is maybe important in studies of the nature, polyethylene glycol, calcium ion, function of plasmodestata, the psychology, polyvinyl alcohol, they are the most and control of mitosis in many multinucleated cells commonly use protoplast fusion inducing a nuclear fusion. There are some of the chemicals which are used in chemofusion. I will again repeat it, and they have different mechanisms. For example, fusion induced by sodium or potassium nitrate is somewhat different. Fusion induced by calcium ion at high pH is somewhat different phenomena. Furthermore, the important chemical which is very important with respect to industry, that is PEG, polyethylene glycol. The fusion with polyethylene glycol is somewhat different. The other mechanism for fusion is electrofusion. Electrofusion is a modern technique of protoplast fusion which involves the use of mild electric field in protoplast. They are the different mechanisms which are reliably used in protoplast fusion. Mechanism of protoplast fusion is very important, and they are a very important explanation which are very important to consider during the study of this topic. Here in this picture given, we can see a simple phenomena of protoplast fusion demonstrated. What is here? There are two type of bacteria cell. One is dark color shade of purple, and other is light shade color of purple. Two bacteria cell having a plasma membrane, chromosome, cell wall. In what we do in these two bacteria cell wall will be digested enzymatically producing protoplasts. There are two protoplasts from two different bacterial cells. These protoplasts will fuse in solution. Protoplasts are treated with PEG, polyethylene glycol. As I have told you earlier, this PEG very important industrial chemical with respect to protoplast fusion. The segments of the two chromosome recombine. Here you can see that recombinant cell has formed having two shades of chromosomes, dark purple and light purple color. Recombinant cell grow new cell wall, and this is how the process of protoplast fusion take place. This is here you can see that this protoplast fusion is by inducing chemicals. What type of chemicals is here is used? Polyethylene glycol. That is, this is here you can say that it is a type of chemofusion example. If we go in the detail of protoplast fusion, protoplasts are formed from bacteria, fungi, yeast, and actinomycetes. When dividing cell are cast to lose their cell wall. Protoplasm may be produced in the bacteria when the enzyme lysozyme. This is an enzyme very important to produce protoplast, especially in bacteria. Protoplast fusion enable recombination in strain without efficient mean of conjugation such as actinomycete. You have seen this phenomena in the picture earlier. It has also been used previously to produce plant recombinant. The technique involved the formation of stable protoplast, fusion of protoplast, and subsequent regeneration of viable cell from the protoplast. So we have two benefit from the stable protoplast. First one is the fusion of protoplast and subsequent regeneration. Another one is the subsequent regeneration of viable cell from the protoplast. Fusion form for mixed population of protoplast is greatly enhanced by the use of polyethylene glycol. We have discussed some other chemical also in this lecture which are used in protoplast fusion. Sodium nitrate, potassium nitrate, calcium ion with the change in pH. Protoplast fusion has been successfully done with Bacillus subtilis, Bacillus megaterium, and among several species of Streptomyces has been done between the fungi geotrichum, and aspergillus. So protoplast fusion is not a new technique. It has been widely used in many industrial processes for the strain improvement to get better results. The method has great industrial potential and experimentally has been used to achieve higher yield of antibiotics through fusion with protoplasts from different fungi. So this is overall a brief overview about protoplast fusion. You have to make the notes of this topic. You should know the proper concept of protoplast fusion. You should know the different methods taking place in protoplast fusion. The other technique which we study today in strain improvement is site directed mutation. First, I will tell you what is site directed mutation. Site directed mutagenesis or site directed mutation, also known as oligonucleotide directed mutation or oligonucleotide directed mutagenesis. It is a technique that can be used to change a single amino acid in a protein sequence. This method involves engineering a DNA primer that contains an altered codon sequence. This primer can then be used along with the original DNA template to produce a new DNA molecule with the altered sequence of the nucleotide that will now code for the protein with the mismatched nucleotide. This is basically we have done site directed mutation at a single point. Sometime what happen in side directed mutation? Sometime we want to slightly alter gene, causing cause we want to slightly alter gene. Why? Because we are not happy with wild type. We want to make direct changes, alter wild type to different gene. Hopefully, the gene will have different function. This is our purpose for side directed mutagenesis. How do we create a gene with alter sequence and in turn alter function? What we do, we got our plasmid. I will explain this phenomena. Please try to listen it with attention. We got our plasmid and we got a normal gene inserted into it. We have the codon that we want to change. For example, this is GAC codon, glycine, adenine, and cytosine codon. In first step, what we will do, create single stranded DNA from it, separate two strand. You can denature it or get virus to do it. Then use synthetic oligonucleotide. Then you will use synthetic oligonucleotide, but this oligonucleotide has a specific base change in it. It has GCC codon, glycine, cytosine codon instead of GAC codon. So this the we have done a site directed mutation at a site where we want which was our target. Oligonucleotide carries that base change you want to incorporate into the gene. Anneal this primer to single stranded DNA and there is one mismatch at one position, but it will still anneal, although not as good as wild type. So there will be some changing in the annealment as in wild type and in the change type. We then add DNA polymerase and dntps, and they will go around the plasmid making the strand duplex. It will make DNA duplex. You also add DNA ligase and it will fix the nick at the end. We got mismatch at one position. This is the phenomenon of side directed metagenesis. Plasmid is intact and it's sealed at the end, introduced into cell through transformation. When this you have now you know what is transformation. When the cell go through replication, some of the copies will look like wild type and the other will be mutant. Then screen and look for the mutants. This is your job. You will screen for the mutants. You can screen for colonies with mutant plasmid, and you do this through sequencing of those plasmid. You can do it with PCR and other techniques too. What techniques you will employ, this depends on you. Mismatch oligonucleotide is key. Once that annealed, complete the plasmid, transfer it into the cell and screen for it. Approximately how long would the oligonucleotide primer need to be? Total plasmid is about 300 base pair in length. When you need to consider what you need to consider, you have to anneal on both sides of the mismatch. You got few bases on both sides, and you need to have you need to have some change in the gene so that DNA polymerase can work. So this is the phenomena by which you can employ side directive mutagenesis. If you see this phenomena in the picture given here, for what is the first step? First step is the plasmid preparation. A gene in plasmid with target site mutation denature the plasmid and anneal the oligonucleotide. Step 2 is temperature cycling. Primers containing the desired mutation using the non-stain displacing the action of PFU turbopolymerase extend the incorporate the mutagenic primer resulting in nicked circular strand. These are basically DNA polymerase. Then what will happen? Digestion. This is the third step. Digest the methylated non-mutated parental DNA template with the DPN1. It's a different enzyme. Transfer the circular nicked of double-stranded DNA into the supercompetent cell, and after transformation, the supercompetent cell repair the nick into the mutated plasmid. This is the basic mechanism of side direct mutation. I have explained this full-fledged concept of side direct mutation in my lecture. Please listen this lecture attentively. If we go in the detail of side directed mutation, the outcome of the conventional mutation is random, but this side directed mutation, the outcomes are not random and they are easily predictable. The recombinant DNA technology and the use of the synthetic DNA now make it possible to have mutation at specific site on the genome of the organism, and this is known as side-directed mutation. That is, you are performing mutation at specific site on the genome of the organism. That you know that what would be the target change. You are making your desired gene, you are altering your desired gene to making your desired product. This is the basic phenomena behind side directed mutation. The mutation is caused by in vitro chain directed at a specific site in a DNA molecule. That all the phenomena is taking place in the lab. The most common method involves use of a chemically synthesized oligonucleotide mutant which can hybridize with the DNA target molecule. The resulting mismatch carrying DNA duplex may then be transfected into the bacterial cell line and the mutant strand recovered. This is the full phenomena which I have explained you in the first slide of side directed mutation. Try to make notes of that concept. The DNA of the specific gene to be mutated is isolated and the sequence of the basis is then in the gene determined. What is happening here? The DNA of the specific gene to be mutated is isolated and the sequence of the bases in the gene determined. Certain predetermined bases are replaced and the new gene is reinserted into the organism. Side directed mutagenesis creates specific well defined mutation. It also helps to raise the industrial production of enzyme as well as to produce specific enzyme. This phenomena of side directive mutation is widely used in industry to form the desired product with the high efficiency, high quality, and low cost production product basically are producing by employing this technique in industry. This is the overall concept about the technique manipulation involving foreign DNA. Thank you very much.